Good afternoon, ladies and gentlemen, to another Worldwide Neurotheory Forum on a Friday afternoon. It's a lovely Friday afternoon indeed in Austria, and I hope you're having a good afternoon everywhere else as well. This afternoon is with us Venkat Ramaswamy from the Birla Institute of Technology and Science in Hyderabad, India. Venkat originally got his PhD at the University of Florida in 2011 and did a postdoc with Chaim Sompolinsky in Jerusalem for 3 years before moving to India, where he did a Simons Fellowship at the National Center of Biological Sciences in Bangalore, and he has taken up a position as faculty, as assistant professor at the Birla Institute just recently, 10 months ago, just in time for the onset of a global pandemic. So lovely time to become faculty, but maybe also just under the line in terms of open faculty positions. Yeah, we're looking forward to his talk. Before I let him start, you know how to ask questions in the right-hand side of your screen. By chat or via the chat box at the bottom of the screen. If they're important, I'll interrupt Venkat. If they can wait, I should say, then we'll wait until after the talk. So with that, Venkat, the stage is yours. Thank you very much for coming.
Yeah, thank you, Tim, and thank you for the invitation and the hospitality. It was wonderful visiting virtually you and your colleagues in Austria and Oxford. Let me just get the share screen working. Okay, so it's thinking about it. Okay, there we go. Let me choose the right window. Okay. All right, are you able to see the slide? Could someone confirm?
Not yet. But
okay,
it'll come. It took a moment last time as well. Hi, Rune.
All right. Okay, I can go back.
There it is. Yeah, I got you. No, I just got to focus.
Okay. So the title of my talk is An Algorithmic Barrier to Neural Circuit Understanding. All right, and the video you see here is that of the Hydra, which was and this whole brain imaging of Hydra from this beautiful paper by Christophe Dupré and Rafa Yuste in 2017. And the things that light up are basically neurons, right, which are briskly active. All right, so there's been extraordinary progress in large-scale experimental techniques in neuroscience over the past decade or two, all right? And by this, I mean things like connectomics, for example, which has almost seen a renaissance and resurgence. And while the first connectome was that of the C. elegans determined in 1986 by Sidney Brenner and colleagues after perhaps a 15-year effort, we now have one other connectome, actually, which is the, in fact, I think one more that came out very recently, but the one before that was in 2016 of the of Siona, of the larval tadpole of Siona intestinalis. And now we have parts of several other connectomes that are coming up. For example, here is one of the Drosophila hemibrain, adult Drosophila hemibrain, which is parts of one hemisphere of the Drosophila brain. This is from the people at Genelia. And parts of others, for example, larval Drosophila, parts of its connectome have been reconstructed. And in fact, even with the fly, there's a move to try to get the complete connectome. And for example, the Wellcome Trust is funding it. And there's even talk of mapping the entire connectome of a mouse. All right, and this was a nice paper, perspective paper that came out on what it would take to do this. And it's also going to cost a fair bit of money by some estimates, a few $100 million. Although some people think it'll cost much more. All right. So in addition to connectomics, in the past decade or so, we've the ability to image neural activity using things like 2-photon calcium imaging has really become very mainstream. All right, and likewise techniques such as optogenetics, thermogenetics, chemogenetics, and these at increasingly large scales in awake behaving animals. All right, and to give you a sense of what I mean by these, I'll just cite a few examples and perhaps take a few minutes to do that. All right, so here's, for example, a paper that came out in 2017, again from a group in Genelia, and what they do here is it's use thermogenetics. So basically, they have 2,000 plus GAL4 driver lines, and so that in a handful of neurons per line, they basically express these TRPA channels. So when you raise the temperature from 25 to 31, those neurons get activated, and then they observe what the fly does when you raise the temperature. And so what they build is a giant behavior to cell line probabilistic map, right? And they do a few other things, but one remarkable thing is the scale of this work. So 400,000 flies were inconvenienced in order to do this, right, which is a large number, and 100 billion annotations of behavior, much of it automatic, and hundreds of terabytes of data, right? And if this seems like a once in a decade work, but and it is in many respects, but just a few years ago, another group in Genelia also did something similar with larval Drosophila, right? But this time it was with fewer animals, but still 37,000 plus individuals, right? Which is still large scale in my mind, all right? And likewise in the act of imaging activity, for example, here's a nice paper from the group of Mark Schnitzer, where they were looking at imaging activity of single neurons, using so the video itself is sort of a wide field epifluorescent imaging. But what they say is that, okay, so I should explain what this is. So the tree-like structures you see are blood vessels, and this is basically obtained by, so the entire dorsal cranium, which is the back of the skull, of this mouse is removed and replaced with a glass crystal skull, and this allows them to image chronically for I suppose months on end. And the tiny dots of light, which you hopefully see, are the single neurons, right? And with this epifluorescent video, I think the estimates are thousands of neurons. But what they say is that if 2-photon calcium imaging could scale to this sort of field of view, then they would be able to access 1 million neurons, which is what the title of the paper suggests. So long-term optical access to an estimated 1 million neurons in the live mouse cortex. All right. That is that. Okay, and indeed, while with the mouse it's not whole brain imaging, in a number of other organisms there has been progress in basically having the capability to image essentially every neuron in the brain. All right, and here is, for example, a video from the lab of Misha Arens, where they image larval zebrafish. All right, so the dots of light you see are individual neurons that light up. All right, the video is sped up a bit, so the temporal resolution is still of the order of a single-digit number of hertz, but this is remarkable, right? So and likewise, people can do so with C. elegans, which is the roundworm, which has of the order of 302 neurons. And here, you can do this while the worm is behaving. So in the bottom left panel, you see the mouse sort of moving through this arena, not the mouse, I'm sorry, the worm moving through the arena. And you can image calcium activity and also infer neuron locations with this sort of technique. And likewise, this is with the Hydra. Again, there's a full screen image of the image I had on the title slide. And again, as you can see, so the Hydra is sort of sandwiched between two cover slips, but it's clearly behaving, it's moving around, and you can also see its neurons light up. And Hydra is especially remarkable because, as I understand it, Hydra are more or less immortal. They can live for extended periods of time, and it's unclear whether they have a set lifespan. And there are other advantages which perhaps I can skip for now. And this is likewise in the adult fruit fly, adult Drosophila, and this is the work of Sophie Amon and others in Carl Deiserot's lab. And here you see a fly moving on a styrofoam ball while whole brain imaging of its brain is being performed. Right? And this uses things like deconvolution techniques and so on. Oops. All right. And okay, so my video stopped. So okay. And not only can people do imaging, whole brain imaging, increasingly people are beginning to be able to do optogenetics and not just things like 2-photon optogenetics and holographic techniques that allow you to stimulate arbitrary subsets of neurons. And people are able to do this while concurrently doing whole brain imaging. And this is a nice paper from the lab of Misha Arens, where they do this in larval zebrafish. All right. So the ability, so not just stimulating all the neurons which have the opsins expressed in them, but also having the ability to individually target neurons and do so concurrently with whole brain imaging. Right. So and there are indeed national initiatives that are aimed at accelerating the development of these and other neurotechnologies. All right. And this includes the United States, Japan, China, Korea, and Australia at this time, and perhaps more are in the works. All right. So really, we are at the cusp of an era in neuroscience, and many of these techniques are being built to largely address the question of trying to understand precisely how mechanistic computation by the network causes the behavior in question. All right. And so again, it feels like we are at the cusp of an era of large scale, and by this I mean including whole brain neuroscience at the network level. All right. And this is the sort of question that we want to get at, or at least many of us want to get at, which is asking precisely how mechanistic computation by the network causes the behavior. And not just is it to understand how that particular individual is doing it, but also even if we are interested in sort of distilling principles that govern how networks perform computations that lead to behavior, you will either need data of this nature to formulate those principles or to verify those principles post hoc once you have hypothesized principles that govern computation. Right? And again, what we would like to have is a fairly comprehensive understanding, right? And so therefore, understanding mechanistic computation in these neural circuits will require efficient algorithms, and which would seek to prescribe the smallest number of experiments necessary that scale as a function of the number of neurons in the network towards this goal. All right. So again, what do I mean by this? What we seek increasingly is not just a correlational understanding where we're recording from neurons and we correlate neural activity with aspects of stimulus or behavior, but we'd like to establish a causal link between neural activity and behavior. Right. And in order to establish such understanding, one requires multiple perturbational experiments. So, for example, optogenetic experiments. And really, it has been unclear exactly how many you need to arrive at understanding, all right, and how that number scales with the number of neurons in the nervous system of the animal. Right? Okay. Now, what is an experiment? An experiment, for example, might entail perturbing activity of a subset of neurons while concurrently imaging their activity and attempting to elicit behavior. Right? And the specifics of the next experiment prescribed by the algorithm could depend on the outcome of the current experiment. Right? And so in this sense, you might think of an experiment as what one considers a trial in experimental neuroscience. Right? So I want to sort of try to quantify how many such trials or experiments you would need to establish a certain kind of understanding. All right? Okay, so what we are doing in a way is to treat the problem of experiment design in neuroscience at the circuit level as an algorithmic problem, and to invoke tools from computer science to this end. And of course, we know from half a century or more of theoretical computer science that certain problems seem to have very fast algorithms, all right, whereas certain others seem to need number of steps that that are prohibitive in the size of the input for that particular problem, right? So some problems are difficult to solve merely because we don't have efficient algorithms for them. Not just that we don't have, that we are able to show that there are no efficient algorithms to solve them in general, right? Or cases where they cannot be solved efficiently, right? So in other words, what I seek to ask here is which class of problems does those corresponding to neural circuit understanding fall in, all right? And in particular, or in other words, how efficient can the most efficient algorithms be to this end? And that is the question. Okay, so okay, so let me sort of give you a brief rundown of the results, so there's no suspense and you know what's coming. All right. So one, I proved mathematically that establishing the most extensive notions of empirical understanding of circuit computation leading to a fixed behavior need exponentially many experiments in the number of neurons in several cases, unless P equal to NP. So this P equal to NP is sort of a technical hypothesis in computer science that is thought to be false, and there's some evidence for it being false, although we don't have a mathematical proof yet. All right. Oops. And furthermore, all right, so this is, of course, the first part is bad news, but furthermore, it turns out that the feasible experimental regime, so clearly nobody can do exponentially many experiments beyond a small number of neurons, but then the question is, how many can I do? What is a feasible experimental regime? It turns out to be one where the number of experiments that you can perform, in fact, scales sublinearly in the number of neurons. Right, so you can do less than linear number of experiments in the number of neurons, and we'll get to this, right? All right. So therefore, the upshot is that determining which notions of understanding are algorithmically tractable in which context, therefore, becomes important. All right. Okay. So what's the sort of broad experimental scenario? So we have an individual animal, let's say, that's been trained to be doing a certain behavioral task, and you'd like to obtain a sort of comprehensive ground truth understanding of how its neural circuits cause the behavior to be manifested at the present point in time, right? And by present point in time, I mean with respect to a single state, right? And so the understanding must be non-correlational in account. Non-correlational, that is, I hesitate to use the word causal for multiple reasons, but what people refer to as a causal understanding, right? And also encompass the degeneracy that's been associated with neural computations, right? We consider behaviors with a binary behavioral readout, right? Which is a way to objectively partition behavior into correct and incorrect. All right. So what does this mean? Okay. So one difficulty in sort of conceptual difficulty in trying to figure out which neurons are involved in which aspects of behavior is the following, right? So let's say the behavioral task involves me picking up this cup of coffee. Right? Now you're going to go in to my brain and let's say silence a bunch of neurons. Now instead of being able to pick it up this way, I go in and I do this, and I sort of, I don't want to spill coffee on my keyboard, but I do it, but not in a particularly smooth way, right? So the question is, at what point do I say that this neuron is actually affecting this behavior? Right? And now, some of you might say, oh, if you're able to successfully do it, then it's fine. Some of you might say, well, if the dynamics of the process changes, then beyond a certain error bar, then you should say that it is not being correctly elicited, and so on. So what I do here in the theory is to say that that is a choice of the experimenter, right? So the experimenter specifies the behavioral readout, right? which is a way of partitioning behavior into correct and incorrect. Right. So after the execution of the behavior, I want a readout which says whether the behavior was correctly executed or not. And this is an issue that's sort of separate from what the theory is trying to address. Right. So having the availability of such a behavioral readout, we then go into to say which neurons, let's say, are involved in the computations and in what sense. Right. Okay. So for example, a possible task could be for a rodent to identify an odor and press a button to indicate the same, wherein our goal is to obtain an understanding of how mechanistic computation in neural circuits causes the animal to recognize that odor A, let's say, and perform the correct behavior to indicate the same. And of course, I keep saying understanding. So Conrad might say, can you operationally define what you mean by understand? All right. And so let's get to that. Right. Okay, so what is understanding, right? So of course, even with understanding, there is no standard definition for what exactly constitutes understanding. Indeed, this is a sort of heavily debated topic if you've been on Twitter at any point of time. All right, and especially understanding of mechanistic computation in neural circuits. And of course, there exist multiple concomitant descriptions that might, example, include details that span different spatial or temporal scales, and depending on one's inclination, one might consider specific kinds of details as important. So then the question is, how does one go about this? All right. So what we do here is we wish to have our analysis apply broadly to many such reasonable notions of understanding, especially ones that span the circuit level. Now, a characteristic of understanding is the ability to answer questions about the subject of understanding in short order. All right. Okay. I'm just checking if I'm on. Okay. Okay, you are. I am. Okay. So a characteristic of understanding is the ability to answer questions about the subject of understanding. Not just answer questions, but answer them fairly quickly, right? So you're not taking inordinate amounts of time to answer it, right? Because you've already understood it in a certain sense, right? So therefore, the most extensive notions of understanding, which I will sort of call comprehensive notions of understanding, ought to enable us to answer certain basic questions about mechanistic computation in the circuit leading up to behavior. Right? And so what I'm going to do here is to posit a list of such basic questions that involve determining certain subsets of neurons that participate in computations leading to this behavior with respect to this behavioral readout. All right, and of course, the intent is not for the list to be exhaustive, but only to demonstrate the existence of a number of questions that cannot always be tractably answered. All right. Okay, so so there are sort of two kinds of settings that that the work talks about. So on the one hand, you might say, you have this individual animal that you wish to understand. Let's say how neural circuits mechanistically cause a certain behavior, and you're running experiments. But the other sort of side of things is, you've already collected a bunch of data, right? So you have like a few terabyte hard disks full of data that you've run over, let's say, several weeks or months, and that's all you have. And then the animal is gone. Now, anything that you need to understand about circuit computation in the animal, you have to sort of distill out from the data, right? So this sort of speaks to both these settings, right? And so so there are two questions. One, what sort of data should you collect, and how does that depend on what you would like to understand? And having collected that data, what are efficient algorithms to then distill answers to those questions? All right. Okay. So before that, a quick segue on notions of reductions in theoretical computer science. All right. So let's say you have two problems, A and B. So, and these are computational problems. They don't have to have anything to do with the brain. So there are two computational problems, right? That let's say your computers execute every day, or your computers would like to execute every day. All right. Now, a reduction from problem A to B is a sort of recipe that, given any instance of problem A, converts it for you to an instance of problem B fairly quickly, all right, in such a way that if you then solve problem B, that you could map the answer back to an answer of problem A, all right. And again, this mapping back is also fairly fast, all right. So so what this shows is that A and B are related in a certain way, all right. And this gives us two things, all right. On the one hand, it tells us that if we had a fast algorithm for B, the existence of this reduction would immediately translate to the existence of a fast algorithm for A, all right. And why is that? Well, you use the reduction to go to B, solve it in B, and then map the answer back to A fast, because your reductions are sort of involve these procedures that are that do not take a whole lot of steps, all right. So that is number one. But more profoundly, they give you one other thing, which is that if it turned out that problem A is in fact very difficult to solve, and let's say you have a mathematical problem that says that there is no efficient algorithm to solve A. Now, it cannot be that B is then easy to solve, right? Because if B were easy to solve, you could always go to B and come back to A and get a fast algorithm, all right? So it would still be an algorithm, right? So it would involve the process of converting the instance of problem A into an instance of problem B, which can be done relatively fast, and then converting the answer back, all right? So so the second implication is that if problem A is computationally difficult to solve, and you reduce problem A to problem B, this implies that problem B is also difficult to solve, all right? And this, in fact, this seems like a fairly simple technique, but this basically underlies a whole lot of important results in theoretical computer science, right? It's a workhorse, the notion of a reduction, which is in this sense. Okay, so what I'm saying is that what I'm suggesting is that with the list of problems that I have, I'm suggesting that those problems ought to be ones that reduce to a comprehensive understanding, right? So if, for example, you claim that you have a certain way of data collection that had all the ingredients necessary for a comprehensive understanding, then these problems need to have reductions to that notion of understanding, all right? Okay, so what's the sort of broad approach we're going to go with? Again, like I said, I'm going to formulate multiple questions about mechanistic computation, all right, that embody colloquial notions of circuits being necessary or sufficient for the expression of a fixed behavior, all right. And I should note here that the use of the words necessary and sufficient in this context have been deprecated. There are a couple of very nice papers that sort of explain why why this has led to a lot of semantic confusion, and that is well taken. But I'm putting it out here just so people sort of at least vaguely understand the sort of thing I'm trying to get at, all right. Now, okay, and okay, so why this sort of a thing, right? So and we have known from roughly a century of neuroscience that not all neurons are involved in all behaviors, or apparently involved in all behaviors, primarily due to things like lesion experiments, where you can lesion or parts of the brain that have been lesioned due to other reasons seem to have only very specific effects on cognition, right? And therefore, which suggests that not all parts of the brain are involved in all aspects of behavior. All right, okay. So having formulated these questions, what I do is I argue that any notion of mechanistic circuit understanding that is comprehensive ought to certainly enable us to answer one or more of these questions in short order, right? Fairly fast. All right, so now I'm going to jump into some definitions, all right? So we can sort of make these concrete, all right? So first definition is one of a degenerate circuit. So a subset N of neurons is said to constitute a degenerate circuit for a behavior B if B can be successfully elicited with the silencing of all neurons except those in N, all right? I'm going to check if...
Okay. Hey, Venkat.
Yeah.
I'm a little late, but someone just asked whether you could give them an example of a reduction in a physical system as an example. That's
okay,
2 slides late, but I just saw the question.
Okay, good. Let me do that. I just checked in to see if I'm still online or... All right, which is good.
You are. I'll be...
Okay, so what is an example of a problem that has a reduction? Okay, how about this? So I'm going to actually go through a specific reduction in this context. So Nico, if that's all right, could you hold on until then? Or I could come up with, so I'm just trying to intuitively,
Sure.
all right? Yeah,
They
okay,
say
good.
sure.
So let me do that. So you will get to see a real reduction. Okay. All right, where were we? Okay. All right, so definitions. Degenerate circuit. So a subset N of neurons is said to constitute a degenerate circuit for a behavior B. If B can be successfully elicited with the silencing of all neurons except those in N. So I'm going to identify a set of neurons, put them in my subset N. I'm going to silence all the other neurons around it. And then I'm going to see if the behavior is still correctly elicited according to the readout that is specified by the experiment. All right. And if it can be, then I call that subset of neurons, I say that that subset of neurons forms a degenerate circuit. All right. Now, there always exists at least one degenerate circuit, which is the entire network, although in practice there likely exist many more. And of course, the presence of a neuron in a degenerate circuit does not necessarily imply a causal role for it, right? Because you could have another degenerate circuit with this neuron removed, but everybody else being the same, which still elicits the behavior. All right. And we've sort of implicitly known of degenerate circuits in neuroscience for long via things like lesion studies, local cooling, pharmacological, optogenetic, etc. Right? Okay, so now getting to the next definition, which is that of a minimal degenerate circuit. So a subset N of neurons is said to constitute a minimal degenerate circuit if that subset constitutes a degenerate circuit, and furthermore, no proper subset of N continues to be a degenerate circuit for B. Right? So I end up with a subset N of neurons, and no matter what way I try to whittle it down, I find that the behavior is not correctly elicited according to the readout anymore. Right? And there exists at least one minimal degenerate circuit, but there could exist multiple minimal degenerate circuits. So if I start off from the entire set of neurons and whittle it down in a certain way, I might end up with a certain degenerate circuit. If I whittle it down in a certain other way, which is to say that I take out neurons in a different order, I could end up with another, in principle, with another minimal degenerate circuit. Right? And in the context of a particular minimal degenerate circuit, each neuron in it is necessary to elicit the behavior, since silencing it abolishes the behavior. Right? Now, okay, so what is a minimum degenerate circuit? Well, a minimum degenerate circuit is a minimal degenerate circuit of the smallest size, namely a minimal degenerate circuit which has the smallest number of neurons. Now, there's at least one minimum degenerate circuit, all right? But there could be multiple ones if they have the same number of neurons, right? Okay. Now, to switch gears, what I might want to do also do is to find a small number of neurons that I can go in and silence that somehow abolishes the behavior. All right. And so to this end, here's a definition. All right. So subset N of neurons is said to constitute a vital set of neurons for a behavior B if that subset has a non-empty intersection with every degenerate set for B. All right. So what I'm going to do is I'm going to build a subset of neurons such that that subset of neurons has neurons that feature in every degenerate circuit. All right. And what this gives me is that suppose I silence this subset, then it guarantees me that no degenerate circuit can be concurrently active. All right, and therefore, the behavior is not correctly elicited according to the readout. All right, and then you can a K vital set is a vital set of size K, and you can also talk about a minimal vital set, which is a subset N of neurons, the minimal vital set, if it is a vital set and no proper subset of it is a vital set. All right, and this I already said. Okay. So the problem setting, okay, so now you might say, well, what experimental infrastructure are you going to assume, right? Can we do optogenetics? Can we do, I don't know, can we just do 2-photon type optogenetics? Can we get activity from all the neurons? All right. So what I'm going to do is to do the most powerful thing in a certain way. So suppose I had a simulation of the entire neural circuit, let's say sitting on my computer, which is exact in whatever sense that you like to the neural circuit itself, and I have the ability to sort of go in and manipulate it at will. So for example, I know the structure of the neuron, I know the connectome, and I have arbitrary access to the neurons in the circuit. Right? So given that sort of access, and starting from an initial state, I'd like to answer questions pertaining to finding sets of the aforementioned type. Okay, so here's a list of problems shown not to have general sub-exponential algorithms unless p equal to np. All right, so one is determine a degenerate circuit of size k, or indicate if none exists of that size. Determine a minimal degenerate circuit of size k, or indicate if none exists of that size. Determine a minimum degenerate circuit. Determine the set of one vital sets, or indicate if there are no one vital sets. Okay, determine a minimal k vital set, or indicate if none exists of that size, and how many minimal k vital sets exist. All right, so these are 6 questions. All right, okay, so I will skip this slide. Okay, but the question this answers is, your simulation setting is nice, but what does it say about the setting when I can just do, let's say, optogenetic? So ask me in the end if you want this answer, right? But I'm going to move on. Okay, so here is, for example, a variant of the first question, which is given a whole brain behavior b and a positive integer k, does there exist a degenerate circuit for b of size k? All right, so this is a yes/no question. All right, and again, I would argue that if you had a comprehensive understanding, you ought to be able to answer this question. All right. Okay, so now a quick summary of what is called NP completeness in computer science. So okay, so now that you know reductions, this should be straightforward to understand. So the class of NP complete problems is a set of problems now numbering in the hundreds, perhaps thousands, which have the property that given any 2 problems in this class, there is a reduction from, let's say you have 2 problems A and B, there exists a reduction from A to B, and there also exists a reduction from B to A. All right, so that is number 1. So every pair of NP complete problems has a reduction between them. And number 2, for none of the NP complete problems do we know of efficient algorithms. So efficient means algorithms that run in time that's or number of steps that's proportional to some polynomial in the size of the input. All right. So this is a set of problems that has been known for close to half a century, since the 1970s, and even before that. And many of them are actually commercially important, and people have been trying to solve them for a long time. Yet we don't have efficient algorithms. And so the existence of a reduction between every pair of problems implies that if there were such an efficient algorithm for even one of them, all of them would have an efficient algorithm, right? And this, if all of them had efficient algorithms, it would mean that the complexity class P is equal to the complexity class NP, right? Which is a bit, which I will not get into any further, right? But this is from theoretical computer science. Okay, so what I'm going to do is I'm going to take one of these NP complete problems. So for each of the 6 questions listed, I'm going to take one NP complete problem and reduce it to that question. All right, and what this implies is that if that question can always be answered in a polynomial number of steps for a certain polynomial, that would imply that every NP complete problem can in fact also have efficient algorithms in that sense. All right, so in a sense, I'm raising the stakes, saying that if your neural circuit understanding question can always be answered efficiently, that would imply that all these other problems in computer science that people haven't been able to solve efficiently for close to half a century would also have efficient solutions. All right, okay, so the other quick point to be made is there is no proof that there don't exist efficient algorithms for the NP complete problems, but it is widely believed to be the case, although there is no mathematical proof yet. It's a big open problem. All right, okay, so here's one such problem. It's called Clique. All right, so if you have an undirected graph, which is just a set of nodes and a set of undirected edges between them, a clique of an undirected graph is a subgraph of it such that every pair of vertices in the subgraph has an edge between them. All right. Okay, so what this means, for example, is that, so here is an undirected graph. Here is a clique of size 5, because among these 5 nodes, you take any pair of nodes and there turns out to be an edge between them. Here is a clique of size 7. All right, and here are 4 nodes that don't form a clique of size 4 because there is no edge between these 2 nodes. All right, okay. So so the clique problem is the question of given an undirected graph G and a number K, to ask if G has a clique of size K, right? And nobody knows of an efficient algorithm that is efficient in the size of the graph and the number K that that solves this problem, that always solves this problem, right? So, all right. So, so what I'm going to do is to do a polynomial time reduction from clique to this degenerate circuit problem, right? So, what I'm going to do is given any undirected graph. Hold on, I'm just going to see if are we still on? Yes. Okay. All right. So, given any undirected graph, I'm going to give you a recipe to convert it into a neural circuit such that the undirected graph has a clique of size K if and only if the neural circuit that I form has a degenerate circuit of size K plus 2. All right. And here's how the recipe goes. So I'm going to take this undirected graph and I'm going to make each node into a neuron and each edge into a bidirectional connection. All right. So, if you're thinking, oh, all neurons don't have bidirectional connections, that's, so I'm going to deal with that case in a bit. Hold your horses. All right. So between any two neurons, if there is an edge, I'm going to put a bidirectional connection. And I'm going to throw in, and these neurons in this corresponding to this graph are going to be what I'll call interneurons. And I'm going to throw in what I call a sensory neuron and what I call a motor neuron. All right. And so there will be edges from the sensory neuron to every interneuron and connections from every interneuron to the motor neuron, right? And there is again a simplified picture where there's only a single sensory and a single motor neuron. And here's what I'm going to show. So first of all, behavior is evoked when the entire network operates, all right? And by behavior, all that I mean is that a single sensory neuron indicates the presence of the correct stimulus, and a single motor neuron spike, I should say. A single sensory neuron spike indicates the arrival of the correct stimulus, and a single motor neuron spike indicates the correct execution of behavior. All right, so behavior is evoked when the entire network operates, so that's number one. Number two, there exists a K plus 2 size degenerate circuit in the above network if and only if the undirected graph that we started off with has a K clique, right? Okay, so like I said, the construction itself, you have bidirectional edges between interneurons, you have a sensory and a motor neuron. Stimulus elicits a single spike from the sensory neuron, single motor neuron spike causes behavior. All right, so this I already said. Okay, so now the conduction delays are the same for each pair of the following neuron types. So sensory neuron to interneuron, the conduction delays are all the same. Interneuron to interneuron, the conduction delays are all the same. And interneuron to motor neuron, the connection delays are all the same. Again, you might say, oh, this is not really the case. No evidence for this being the case in the brain. Fine. What I'm going to do eventually is to embed this network in a bigger network where it seems that this is not the case. All right. And I'll get to that in the next slide. So the spike from a sensory neuron to an interneuron elicits a single spike in each interneuron. So the sensory neuron to interneuron synapses are really strong. So a single spike from the sensory neuron causes each interneuron to spike. So that's number one. Number two, if all my interneurons spike concurrently, the motor neuron spikes. All right. Number three is that if there is a pair of volleys of spikes from a set of K interneurons, the motor neuron just about spikes. All right. And in particular, spiking from a set of less than K interneurons concurrently can in no event cause the motor neuron to spike. Right? So that's the next condition. Right? So what this therefore does is that if an interneuron is not receiving input from a sensory neuron, it needs concurrent spiking from at least K minus 1 other interneurons to produce a spike. All right, so this is an additional condition. So if an interneuron is not receiving input from the sensory neuron, it needs concurrent spiking from at least K minus 1 other interneurons to produce a spike. So what this does is that the second volley of size K is only activated if there is a K clique in the underlying undirected graph. All right. Okay. So certain other remarks. Okay. So one, the interneuron to interneuron connections were bidirectional, and all interneurons were excitatory. All right, which is also important, but this is not really necessary. So what I can do is I can introduce an arbitrary number of auxiliary interneurons with the constraint that each of them may receive connections or send out connections to at most k-2 other interneurons, all right, and the inhibitory ones don't send connections to the motor neurons, all right. So in other words, I'm embedding this network in a larger network such that, you know, your let's say excitatory neuron to inhibitory neuron ratio is consistent with known experimental results and so on, right, and the results and the connections don't seem bidirectional very often and so on. All right. So, okay, we're still on. So what I've shown is I've shown a reduction from Clique to Degenerate Circuit. I just did, all right. And I, by definition, I've argued that there exists a reduction from Degenerate Circuit to any notion of comprehensive understanding. All right. So, so there are 5 other problems with reductions, which I'm going to skip in the interest of time. Okay. So now there are caveats with this, right? So this whole enterprise of NP hardness, which is the act of proving such reduction results, is a worst case result. All right. What does that mean? It means that the results show that there exists a subclass of neural circuits which provably require exponentially many experiments unless P equal to NP. All right. In other words, not every circuit. This does not establish that every neural circuit requires exponentially many experiments, but it says that there is a certain class of neural circuits in this case. All right. But worryingly, you know, if you paid attention to the reduction, the class of circuits in the reduction is a rather simple class of circuits that just maps stimulus to response, while sort of being mediated by this cloud, which is your interneuron circuit, right? And so, on the one hand, it is natural that one might expect that circuits doing more sophisticated sorts of computations might be at least as hard to understand comprehensively. Right? And also, analysis is for a single state and a single behavioral readout. Right? Now, one could argue that, to really understand circuits, you will have to look at multiple states and multiple behavioral readouts to really get at how the circuits are performing computations that lead to behavior. The other issue is the temporal aspects of participation of neurons is not treated here. In other words, I say that a neuron is either required or not required during the course of the trial. Now, it's possible that it may only be required in the first half of the trial, not the second half, and so on. So those aspects I'm not considering, and if one does consider those aspects, that increases the your state space in a way. Right? And the analysis is restricted to the acute case, and chronic is different. And I'm going to not say anymore, although I could. And in particular, I want to emphasize that this is not an impossibility result. All right. This doesn't say that for every neural circuit you cannot do this. So it's a worst case result. Namely, what it is saying is that if you had hoped to have an algorithm to answer any of these 6 questions that would always finish efficiently, then no matter what the neural circuit, then you are out of luck. Right? Okay. So this is fine. So exponential is horrible, and everybody knows it. All right, requiring an exponential number of experiments in the number of neurons. Exponential functions grow really fast. But the other question is, well, how many experiments can we do in practice, right? So to obtain a rough upper bound, I'm going to consider, well, how many experiments can you do in the lifetime of an individual animal, all right? And how does that compare to the number of neurons in it, right? So to this end, I'm going to assume that each experiment takes 1 second, all right, which is corresponding to the behavioral time scale, and conservatively that there is no gap period between successive experiments. So I'm going to basically run my circuit interrogation experiments from cradle to grave of my individual animal, all right? And so what I did is plotted estimates of the average lifespan of a number of organisms in seconds, which is also a proxy for the number of experiments one could perform in an average lifetime, given this assumption. And I plotted that number as a function of the estimated number of neurons in their central nervous system. And this is what you see. Right? So what you see is a log-log plot of estimates of the average lifespan as a function of estimated number of neurons in the nervous system for C. elegans, fruit fly, mouse, octopus, marmoset, human, chimpanzee, rhesus monkey, and elephant. All right. And those are the dots in blue. All right. And the sort of cyan line is the y equal to x line. All right. So what this says is that typically you can do less than linear number of experiments in the number of neurons of organisms, right? Although there are notable exceptions. I think one of these is C. elegans. I don't remember what this is, although I can look it up. In other words, what this is also saying is that most animals are estimated to have more neurons in their nervous system than the number of seconds they live, which is something you may not have thought about, and which I didn't either before I did this. All right. So now you might say, oh, why wait for the entire? It might seem that the limiting thing in this is the behavior itself, which takes a long time. So why wait for the entire behavior to be executed, right? And there's a beautiful paper from the 90s where they said, for example, there was evidence that speed of visual processing in humans is of the order of 150 milliseconds, or in certain kinds of discrimination tasks or categorization tasks. So why do we need to run the entire trial? We're going to just find a bunch of neurons that are correlated with the trial being correctly executed, and when we see those neurons spiking, we're going to call it a day and abort the trial and call success. All right. So suppose we had neural proxies of behavior that manifested in 100 milliseconds. So I replotted the same graph with each experiment basically being of 100 milliseconds instead, and you find that it doesn't really change things dramatically. Things are still stuck around the y = x line. All right. So we are still stuck around linear or less than linear, even with this kind of an advance. All right. So I'm almost at the end. So the results here, to summarize, suggest that in addition to current technological impediments, there exists another fundamental barrier, which is an algorithmic one to neural circuit understanding. Namely, it is the idea that there may not exist efficient algorithms that can be used to interrogate neural circuits to infer computation in them, or to understand computation in them. So that's the idea. That's the algorithmic barrier, as I call it. All right. And therefore, going forward, causal understanding of neural circuits is likely to be frequently driven by algorithmic constraints rather than limitations in experimental methods. All right. And specifically, theory agnostic neural circuit interrogation might end up being onerous in many cases. All right. And theory is sort of a natural way to constrain the set of possibilities in these sorts of settings, where you use a theory to guide the sort of experiments that you might have to do. All right. And especially in the past decade or so, with experimental technology improving so much, one view has been that because we can measure so many things, perhaps theory is not as important. Because in the old days, a theory was very important, because if a mouse had 75 million neurons and you could only record from 100 of them, it was thought that theory was very important to sort of interpret what those 100 neurons or handful of neurons were doing. But what I argue here is that even with the ability to record from every neuron, theory will have an important role to play in shrinking the space of experiments that you could possibly have to do. Now, two directions to take. One could ask, ask questions for which there exist algorithms that are always guaranteed to finish, for example, in the sublinear number of experiments in the number of neurons. So in other words, sort of drop this requirement of trying to comprehensively understand or ask any which question you want and have the expectation that you'll be able to always answer it. Just focus on simple questions that have algorithms that are always guaranteed to finish. So that's one possibility. Two, the slightly more than slightly more difficult possibility is investigate which subclasses of neural circuits can be understood in a sublinear number of steps, even if they cannot be generally understood in such a small number of steps. And like I said, theory looks like it'll have an important role to play in this direction. All right. So there are also examples of questions that can be answered rather fast, which I won't have time to get into. But for example, there's a definition of a quasi-minimal degenerate circuit, which can be whose question can be answered with log n number of experiments, where n is the number of neurons, and log n grows. The logarithmic function grows very slowly with n. So even if you had 100 billion neurons, you would need something like 37 experiments to answer this question, which requires log n number of experiments. All right. So it's sort of, this is close to my last slide. So neuroscience, especially with vertebrates at cellular resolution, has largely been correlational so far. And given the sort of breathtaking advances in experiment recently, we need systematic theory to define conceptually what causal notions of mechanistic understanding are. All right. And again, so for example, deep learning is going through a similar sort of crisis, if I may use that word, wherein we have these wonderful deep networks that are able to solve these problems with extremely high degrees of accuracy, yet we don't have a satisfactory understanding of the computational principles that govern them. All right, so neuroscience is not alone. Okay, so the point was that even with the systematic theory, we will have to bake in algorithmic tractability into it in order to ensure that our experiments will finish. All right, and really the big open question is, even with this algorithmic barrier, to what extent is it going to be important, and can we really circumvent it by the sort of ways that I talked about in the previous couple of slides. All right, and so this is the take-home message, and a preprint of this is up on bioRxiv, and I am happy to take questions. Thank you.
There you are.
Okay.
Sorry, I'm not on my own computer, so I had a little trouble navigating the mouse.
Yeah.
Thank you very much for your talk. We do have a few questions. Before I read anyone else's questions, I'll ask my first own question. I have two. Number one is, you don't mention him explicitly, but in Mars levels of understanding, I assume that you're always referring to implementational understanding. So how is anything implemented? And I was wondering if you think the same barriers exist for algorithmic or even only computational understanding, or how that would change the scenario.
Right.
Maybe that's my first.
So, so the sort of short answer is I don't know, or I don't have more than a superficial answer, but I'll give you the superficial answer. And the superficial answer is that several parts of AI, notably computer vision, have been premised on the fact that there is a clean separation between the implementational level and the computational level. So, for example, computer vision, for about 30, 40 years, maybe 40 years, has been premised on the fact that there are algorithms for computer vision, that the brain uses, and then there's implementation, and we don't care about the implementation. Let's go after the algorithms. And beyond a few notable exceptions, computer vision hasn't been successful before deep learning. And a lot of computer vision folks now think that this was a mistake, namely that maybe there is no clean separation, that maybe the algorithms don't exist independent of the neural substrate. And so, but that could also be wrong, of course.
So does that also speak for Mars' overall assumption or separation being problematic, or is that just for this specific problem?
Right. So I think it is unclear whether the separation exists. All right. Now even with Mars, other levels, for example, there are critics of it, as you know, Jonathan Pillow being a notable one, where people feel that they are perhaps misguided in different ways, but maybe that's a topic for a different time.
All right. Yeah. Another question I had was, you're speaking of, you say you're using very simple tasks, or you're using one protocol, and as simple a protocol as you can imagine. Do you think that you could reduce the amount of degeneracy, or reduce the amount of alternative interpretations if you make the task harder? So if you do the hardest task in the shortest amount of time, would that change? Right. Or would that change your assumption?
Right, it's entirely possible, and again, I don't have a definitive proof, and again, this is sort of an intuition that people have, namely that more difficult tasks only have a few possibilities of solving them correctly, and so it could well be true. So again, what I said was also speculation in a certain sense.
Okay.
So yeah.
Then we have a couple of questions from the audience. Right. Have you tried this approach for different kinds of questions other than finding degenerate sets? And do you have an intuition about what are the properties of the problem that make it impossible to find an efficient algorithm for?
So, so for the other 5 questions, I have similar reductions, although they are not identical. What properties of the problem make it impossible to find an efficient algorithm? So again, so this also goes back to, I think there is no clear sort of answer that you can demarcate, and this is in fact even true with computer science, where there exist problems that look very, very similar, yet one of them has a fast algorithm and the other does not, right? And one can go in and look at the proof, but it's often not very clear what exactly draws the line between the two. All right. Not a satisfactory answer, but
fair enough. Someone asked a slightly more, I should say, fluffy question: whether you could do anything with quantum computation techniques.
Well, I don't know. All right. Also, I don't know enough of quantum computation to answer it meaningfully. But having said that, I think the one bottleneck is also behavior being so slow. Although even if you reduce behavior by an order of magnitude, it doesn't really affect things much. But in other words, so if you try to parallelize the process, you still have to have a sense of how behavior is being evoked. So in that sense, it seems like it's not parallelizable. But of course, it is open to sort of clever ways of looking at it, and who knows, maybe that could work. But quantum, I'm not. I thought the brain was classical, no? I don't know. Yeah.
I don't know either.
Yeah. Okay.
Do you see any hope for solving the brain?
Whoa. Okay. Again, so I want to. Okay.
Talk, right? Other
What's
than
that?
You're giving a pretty pessimistic talk all over.
I know. Okay. So, all right. So let me. Okay. So the answer to that is that maybe this notion of drawing lines around subsets of neurons and asking which is a necessary circuit, which is a sufficient circuit, are perhaps the wrong questions. As you said, the really meaningful question is the one about what is the algorithm, or what are the principles, or what is common to various implementations. And many of these questions are sort of digging into the specific implementation and asking questions about the implementation. All right. So that is point number 1. All right. Which is the sunny view of things. And point number 2 is the pessimistic view, which is saying that look, there are questions about Turing machines which are undecidable. All right. So Turing machines are things that have a small encoding, all right, and it's basically completely specified. Yet there are questions, in fact, your halting problem type questions, but there's a larger body of questions, basically, which include things such as where you're trying to, informally speaking, debug your programs and so on. So those kinds of understanding Turing machine questions, a lot of them are undecidable, which means that you cannot have one general algorithm that in fact even answers the question given arbitrary amounts of time. So that is the pessimistic answer, but that also points to the fact that you could construct specific different algorithms for restricted subsets of subclasses of circuits in this case that could do the job. So I think the take-home message is that this is a problem that needs more attention, especially in the neuroscience setting, where we have to think about what does it conceptually mean to understand, and do these sorts of algorithmic barriers exist for other kinds of understanding that we desire. All
right. I think that's it for today, anyway. Thank you very much for coming, Venkat. Thank you.
Thank you. Yeah. Thank you, Tim, and it was a pleasure. And thank you for the.
Maybe for those of you who are still around, there'll be a learning salon tonight, so tune in again. And otherwise, I'm not sure we're going to have a Neuro Theory forum actually next week, because we had a cancellation, but stay tuned. We might get a substitute. So have a lovely weekend and see you
Thank
later.
you. Take care. Take care, everyone.
Bye.